DNA replication is the process by which DNA makes a copy of itself during cell division, creating more DNA. Okay, so the first thing that we need to know is DNA replication happens right here in this part of the cell, also known as the nucleus. When the cell replication happens. So cell replication happens during a phase called interphase. So interphase is when a cell is growing, when it is carrying out cell processes, and it is replicating its own DNA. The first step in the replication of DNA is unzipping the double helix structure of the DNA molecule. Helicase is an enzyme used to help the process by breaking down the hydrogen bonds that are holding the complementary strand of DNA together.
The separation of the two single strands of DNA creates a Y shape called a replication fork. The two separate strands act as a template for making the new strands of DNA.
One of the strands is orientated into the 3' to 5' direction, which is towards the replication fork. That is called the leading strand. The other strand is orientated into the 5' to 3' direction, which is away from the replication fork, and that is called the lagging strand. As a result of their different orientations, the two strands are replicated differently.
The leading strand is a short piece of RNA called a primer, which is produced by an enzyme called primase. This comes along and binds together the end of the leading strand. The primer acts as the starting point for DNA synthesis.
Chunks of DNA called Okazaki fragments are added to the lagging strand in the 5' to 3' direction. This replication is called discontinuous, as the Okazaki fragments will need to be joined up later.
DNA polymerase binds to the leading strand and then adds new complementary nucleotide bases like A, C, G, and T to the strand of DNA in the 5' to 3' direction. This replication is continuous. Once all of the bases are matched up, A with T, C with G, an enzyme called exonuclease strips away the primers. The gaps where the primers were are then filled by yet more complementary nucleotides. The new strand is proofread to make sure there are no mistakes in the new DNA sequence.
Finally, an enzyme called DNA ligase seals up the sequence of DNA into two continuous double strands.
Following replication, the new DNA automatically winds up into a double helix. Now I'm going to draw the DNA double helix with the sequence on the 5' to 3' strand. Okay, so I'm going to start off with A right here, C, G, A, G, T, A, and C. Okay, now on this strand, which will be the 3 to 5 strand, I'll be writing the complementary bases. So this one will be T, G, C, T. The first step of DNA replication is helicase. Which begins to unwind the DNA at the replication fork.
Okay, now that it's unzipped, the DNA polymerase come in and adds a complementary strand to this one, making a 5 to 3 leading strand. Okay, now that we have the complementary strand from the 5 to 3, you now want to go into the 3 to 5, 3 prime to 5 prime direction. In this direction, it forms Okazaki fragments on the lagging strand, which is this one is the lagging strand, and this one is the leading strand, because this would be complementary, and they are joined by DNA ligase, the glue. Now that we have our complementary strand, ligase would come in making a double helix with new DNA fragments. Now, what we need to remember. One, that helicase, it unwinds the DNA strand. Two, protein binds DNA. Three, primase, it creates a primer. Number 4, DNA polymerase, it builds a DNA strand. And 5, ligase, it glues fragments together, creating another double helix strand.